Hello, this is a non-enveloped DNA virus virology tutorial presented by the SchrDbb. We reference Lippincott Illustrated Review of Microbiology by Cynthia Cornilesson, Bruce Fisher, and Richard Harvey. So here we're going to see non-enveloped DNA viruses. So with the non-enveloped DNA viruses, we're going to first start with the classification of non-enveloped DNA viruses. So in the classification, you're going to start first with DNA viruses. We said that there are three main parameters which are helping you in order for there are four main parameters to five main parameters helping you to give the classification of viruses very easily. The first thing is the genetic. There are two main types of viruses based on the genetic material. They can be DNA virus or they can be RNA virus. That's the first parameter. Now the second parameter is to know if the DNA is single or if the genetic material is single stranded or double stranded. That's the second parameter. The third parameter is to know if it is single stranded, generally with the RNA. So if it is single stranded, generally with the RNA, you have to know if it is positive strand or it is a negative strand. So that is the second parameter. So the first parameter is single stranded, double stranded. Third parameter, the third parameter is it a single? If it's single stranded, then is it positive strand or negative strand? The fourth parameter after the genetic material is it having what is the symmetry type? Is it icosahedral or is it helical? That's the fourth parameter. And then the fifth parameter is if it is envelope or non-envelope. So those are the five main parameters when you want to deal with the classification of DNA very easily. So we start with the first case. So in this first case, you have DNA virus. The DNA virus can be divided into two. We have single-stranded, double-stranded. Now this one, it is single-stranded non-envelope. Example of single-stranded non-envelope, we have Parvoviridae, which is a family, Viridae for family. So example of this Parvoviridae, we have Parvovirus. Virus just means that it is a species. Is it clear? So when we put the word virus underneath, it means that it is a species. So now the next one is the double-stranded. So for double-stranded, we have envelope, non-envelope, and we have envelope. Is it clear? So under the non-envelope, we have the Papilloma and we have the Adenoviridae. So example of Adenoviridae, we have adenovirus. An example of Papilloma, we have Papilloma, and we also have Papilloma viridae. Under Papilloma viridae, we have the human papilloma virus, and we also have the Polyoma virinae. Under the Polyoma virinae, we have the BK virus and we have the GC virus. Is it clear? Viridae is for family, and virinae is the subfamily. Is it clear? Now, so this diagram is just to tell you that this is single-stranded and it is red to show there's DNA. This is double-stranded and linear DNA, and this is double-stranded circular DNA. So now let's visualize now the relationship between the steps in the development of the skin wart and the life cycle of human papilloma virus. So you need to know that. So these were examples of the sorry for that. These were examples of the different non-enveloped DNA viruses. So generally, the non-enveloped DNA viruses are divided into two. We have the Papillomaviridae and we have the Adenoviridae. Those are the two examples of non-enveloped viruses. So under this, we have now two. So under the Papillomaviridae, usually the Papillomaviridae are having a circular double helix DNA, double-stranded DNA. While the Adenoviridae are having a linear double-stranded DNA. So now under the subfamily, now we have the Virinae of the Papoviridae. We have the Papilloma Virinae. So example of Papilloma Viridae, we have human papilloma virus, and under the subfamily of Papoviridae, we have also the Polyoma Virinae. So example of Polyoma Virinae viruses, you have the BK virus and you have the GC virus. So we are going to work here with the human papilloma virus. So this is the step for formation of the skin warts from the human papilloma virus life cycle. So at first it starts with initial infection of the basal layer of the cells that is going to cause a multiplication and spread laterally in addition to the daughter cells. Is it clear? So what happened is that it's first going to start in the viral life cycle. So normally what happened is that we have the basal cells which are going to divide to produce the stratum spinosum, and then we are going to produce the stratum corneum. Is it clear? But what happened is that when the virus is going to be affecting, so what happened is that you have replicating viral DNA is going to express their early gene in the viral, so it's in the microorganism. So we are going to have the division. We have replication of the replication cycle begin with the expression of the viral early genes, followed by the multiplication of the viral genome, and finally the assembly of the viral progeny at the superficial layer of the wart. As easy as that. It just infect the skin and then they start multiplying. Now we have two main types of wart location. You can have cutaneous wart and you can also have mucosal wart. So for cutaneous wart, it's most common wart and it is benign and resolves spontaneously. Is it clear? So that's the cutaneous wart. Most common form of wart is cutaneous, but it's benign and can resolve spontaneously. In genetically predisposed individual, warts can lead to epidermal dysplasia and squamous cell carcinoma. So generally, you need to know that what may be due to a virus such as papilloma virus infecting, but what can also be, so that papilloma virus can occur also in genetically predisposed patient, or the what can occur on itself. Is it clear? But generally associated with human papilloma virus in patients who are genetically predisposed, and other patients who are also genetically predisposed are also capable of having epidermal dysplasia and certain forms of squamous cell carcinoma with those warts. Now the next one is the mucosal infection. So a certain mucus can be affected with warts. So like example, we have laryngeal papilloma is benign but can be affected with warts. We can have that at the level of the larynx. You have anogenital warts which can occur at the level of the vagina and the anus, is usually also benign. And we have infection of certain type of papilloma virus can also lead to cervical carcinoma. Example, human papilloma virus 16 and papilloma virus 18. While the other forms of human papilloma virus, such as human papilloma virus 6 and 11, is only going to cause this mucus and cutaneous warts, which is not going to cause any carcinoma. So these are certain forms of example of warts. So example of cutaneous warts can be seen here. This example of cutaneous wart. This is a flat wart here, and this is an adult genital wart. And don't forget that there is also a syphilitic wart that can be produced at the level of the anus, which has no relationship with the wart that is produced from human papilloma virus, which is usually a finger-like progression. You need to know that there are three main types of wart. You can have a condyloma acuminata. So condyloma acuminata is the one that is going to be finger-like projection. We have for condyloma papularis, it is the one that is going to resemble like a papule, and it is flat. And we have another type of condyloma, which is also another form of wart. So now, what are the treatment of the wart? So for the treatment of the wart, you can use surgical removal with a knife or with a surgical blade associated. Is it clear? So you have destruction of wart. The destruction of wart can be done by certain product, cytotoxic products such as nitrogen. It can also be done with laser vaporization. It can also be done with cytotoxic chemicals. So example of cytotoxic chemicals, we can have ethanoic acid, which can be an example of a cytotoxic chemical. We can have povidone, we can have other chemical, so which is used in case of what you have drug also used, can have interferon for laryngeal papilloma. Now, what are the location and properties of papilloma virus infection? So don't forget that there was papilloma virus, and the other class was containing of the BK polyoma virus and the GC polyoma virus. Now, in case of the GC virus, you need to know that GC virus is going to result to the disease called the progressive multifocal leukoencephalopathy. Is it clear? So in progressive multifocal leukoencephalopathy is a rare fatal demyelinating disease caused by GC virus, and usually the patient that have GC virus is usually an opportunistic infection with HIV virus. Is it clear? So when you have HIV virus, it predisposes you more to have the GC virus, because you know that HIV virus is going to affect the DNA genome of the patient. Is it clear? So when you have already that predisposition to the DNA genome being affected, you have again GC virus being entering into your body, you're going to can have this fatal demyelinating disease of the brain. It can be fatal demyelinating disease of the brain. So in this demyelinating disease of the brain, what happens is that you have removal of myelin sheath from the brain. You have loss of the part of the central nervous system which is myelinated. So it's called progressive multifocal leukoencephalopathy. Multifocal means that it can occur at the different parts of the brain, and progressive because it takes its time for it to occur, and usually occur in immunocompromised patients, I've just said. Now the next is for the BK virus. So BK virus is hemorrhagic cystitis is a disease that is caused by the BK virus, and commonly seen in immunocompromised patients also. So you have a patient who is immunocompromised having HIV, and then that patient is passing out asymptomatically asymptomatic hematuria. You just the urine he's having urine is only is reddish, that's hematuria, and also having symptoms of irritation of the bladder, such as cystitis. So that is the BK virus. So on a radiograph, on an electron microscopy, you can visualize the BK virus as shown here. Is it clear? But usually that is not used in our context. So this is another way to visualize the different viruses. So these viruses now are icosahedral in symmetry. Is it clear? So this is an electron microscope now to visualize the adenovirus, and the adenovirus is going to be icosahedral in symmetry. Now, you need to know that the infection caused by adenovirus, you have multiple infection caused by adenovirus having the adenoviridae family. You can have ocular infection. For ocular infection, it can occur in cases of follicular conjunctivitis. You can have follicular conjunctivitis with adenovirus. You can have keratoconjunctivitis with adenovirus. You can have respiratory infection. Respiratory infections such as acute febrile pharyngitis, pharyngoconjunctival fever, acute respiratory disease, and you have viral pneumonia. Is it clear? So you have acute febrile pharyngitis, pharyngoconjunctival fever, acute respiratory disease, and viral pneumonia, all that occur in the case of adenovirus. Now we have gastrointestinal infection such as infantile gastroenteritis can occur with adenovirus, and then the last infection with adenovirus is also a hemorrhagic cystitis. Now this is a replication of the parvovirus B19. Is it clear? Now the parvovirus B19 was not inside the envelope double-stranded DNA. It was instead the single-stranded DNA, non-enveloped double-stranded DNA. This parvovirus was classified up there inside the single-stranded DNA. Is it clear? So the non-enveloped single-stranded DNA. So among the non-enveloped single-stranded DNA, we have now the parvovirus with the under the family Parvoviridae. So this is the replication of the parvovirus. Is that the virus is going to attach and penetrate the host cell. That's the first thing. Then the second, you have DNA genome is released into the nucleus. Usually, you need to know that the parvovirus is going to affect the bone marrow. So mostly, all of this is going to occur inside the bone marrow cell. So it attaches, and then the DNA genome is going to be released into the nucleus. Now you need to know that what parvovirus, it is a single-stranded DNA. With a double-stranded DNA, it's very easy for them to affect the host cell. Why? Because it's going to directly enter the DNA of the genome, the host cell. Is it clear? It's going to be integrated with the DNA of the genome, and the enzyme that is going to be helping into the integration into the DNA of the genome is going to be called the enzyme integrase. Is it clear? Now, in the case of a single-stranded viral DNA, so what happens is that you are going to have a replication first by the DNA-dependent DNA polymerase. So when you have a replication with DNA-dependent DNA polymerase, when I say DNA-dependent DNA polymerase, I just mean that what it is DNA-dependent, meaning that it is using DNA to produce DNA. That's DNA polymerase, the next thing coming. So that's the replication. So it's now going to replicate now double-stranded DNA. Is it clear? Now that double-stranded DNA now is going to be integrated into the nucleus. That the enzyme integrase. Is it clear? When it's integrated now in the nucleus, now the host RNA polymerase now is going to produce the messenger RNA that is going to move out of the nucleus now to be transcribed into structural proteins. Is it clear? Also, you have now the non-structural proteins now can also be helping. The non-structural proteins which were first produced are going to enter back and are also going to help in the replication again of that double-stranded DNA to produce the messenger RNA that is going to produce again now structural proteins. Now the structural proteins now are going to bind now with the viral DNA to produce again the nucleocapsid that is going to be released. Is it clear? So this is just the parvovirus B19. So we have not seen this cycle. So we have ended at type 4 with the RNA viruses that is going to continue to produce the DNA cycle. Is it clear? But this now is type. This we have ended with type with type 4 that was having that. Now with type 5 we have a single-stranded DNA virus that is going to produce as such. But this DNA virus is much more easier to understand than RNA viruses. Now, this is a typical slap cheek appearance in a child that is infected with parvovirus. Is it clear? So it's as if you have slapped the jaws of this child. So it's a slap cheek appearance in a child that is affected with parvovirus. So generally those were all that. Now we have other diseases in cases of parvovirus. Can have erythema infectiosum, we can have bird defect, and you need to know the laboratory investigation of the parvovirus is it's not routinely done. That's the first thing, but you can use DNA detection methods like PCR, polymerization reaction, which can be helping you to visualize here. And here for the treatment, no antiviral agent generally is used for parvovirus. Those are the things that to visualize here. So from here we are finished with this part of the tutorial. Say thanks for your kind attention. Please don't forget to like and subscribe for our channel, Sound User Makers. Thank you.